This is Shelley Kraft coming to you live on SNNLive. We're at the Disruptive Growth Company Showcase 2017 here in New York City. Standing with me today on the program is Richard Schumacher from Pressure BioSciences, Inc. It's a publicly traded company on the OTC markets, and the symbol is PBIO. Rick, welcome to SNNLive again.
Thank you for having me on. Really appreciate it.
It's good to have you back.
Thanks.
Let's get started with an overview of the company, and then we'll go from there.
Sure. Well, we are a life science tools company. We make instruments and consumables that are sold to the research market all over the world. We have about 275 instruments that are already placed. We did $2 million in sales last year in 2016. And what's neat about that is it's the older instrument. We now have a brand new instrument. And in addition, we've hired a sales team. Shelley, you've known me for years. We've had one person in sales for all those years. We now have 6. So I'm excited about that because the sales team is now out in the field, and we're looking forward to ramping up our sales.
Okay. So let's take the next question is, tell me what kinds of customers you sell to.
We sell to the researchers around the world that you hear about every day. When you listen on the radio, watch TV, you hear about the researchers coming up with a new drug, a new cure, a new compound that might help solve an issue, maybe addiction or maybe cancer. So we sell to them an instrument that they use to break open the samples. Remember, the information we need is in the molecules inside cells. It's in the DNA, the RNA, the proteins, the lipids. How do you get to those? We're at the molecular age. We need to break those cells and get those out, and then there's as many as 10,000 different proteins and maybe 1,000 different lipids in that cell. We need to break them open, get those biomolecules out, and then study them as researchers. That's what we do. We do it in a whole new way. I'm a scientist. 20, 30 years ago, I did what people do now. We break the samples like breaking a walnut with a sledgehammer. We just do whatever we can to break that cell. What we do as a company is we treat it like a sponge. We squeeze it back and forth. We use pulses of pressure cycling technology, our patented technology. We squeeze things out of cells. It's far more higher quality, far better, and we've now brought on a number of the key opinion leaders around the world that are starting to use our technology.
I think of your technology as something that would be placed in labs at colleges in their bioscience area. Is that where you would see your equipment most?
Yeah, we are in many labs in colleges and universities. We have about 165 customers now. And we have about 300 instruments placed, which means some of our customers have 2 or 3 instruments. Again, they're using us to break up samples, human, animal, plant, microbes. They're using us to break up normal or diseased or cancer samples because they're trying to get out the molecules inside. So and to study them and find the next cure, find the reason that someone cannot accept a certain therapy and other people can. Why somebody gets a disease and somebody else doesn't. How do we cure a disability? It's coming from the information inside the cells. So it's the researchers on the campuses of colleges and universities around the country and around the world. It's researchers in the government. It's researchers that are at the pharmaceutical and diagnostic companies around the world. These are our customers.
It sounds like, you'll pardon the expression, a microscope on steroids.
Well, that's a good expression. We are at the molecular age. We're doing amazing things with the technology we have, but it all starts by how you prepare the sample. You may have an answer, a cure for a disease inside of a cell. When you look at 10,000 men with prostate cancer, and you know that 300 or 400 passed away, died of cancer, and the others survived, you're trying to find the reason. What's the difference with the cancer with these 300 versus the other 9,700? So you look at all 10,000, you break the prostate cells, you break normal and cancer cells, looking for what's different. And once you find what's different, then you can start looking for a cure. That's what we do. We go and we sell this instrument as a new way of breaking cells that we think higher quality will give them a higher chance for discovery.
Are you the inventor? And let me ask you a question. Why did you think there needed to be a new way to do this?
That's a great question, Shelley. I'm glad you came up with it. We did it because there is a need to extract these biomolecules from cells in a way that's better than what's being done, in our opinion. I'm a scientist. I spent 13 years at Harvard Medical School doing research, and I know that I was taught to break a cell by just spinning it with beads and beating it up until it's broken and we let the insides come out. That is not a high-quality way of doing it, but it's the number one method being used in the world today. It's called a bead beater, and we use sonic waves and we use propeller blades to break cells. We decided that a new method, discovery starts with how you prepare the sample. You need to have high quality with how you prepare the sample. So it was a definite need out there to have a higher quality way of breaking a cell so you don't damage the things you're getting out of the cell that you're studying.
Is it more consistent than all other methods?
Great point. It's more consistent, it's more reproducible. In science, reproducibility is key. What we want to do is, if we find something that works, we want to do it exactly the same way every time. If you're spinning a cell with a bunch of beads, you're getting what you get every time. If you put it in a pressure chamber and you dial in the pressure, the amount of pressure, and how many times you're going to cycle or turn the pressure on and off, how many times you're going to squeeze that cell, you can do it exactly the same way every time. So yes, that's why a number of key opinion leaders in proteins and DNA all around the world have switched to our method in the last several years.
How big a market is it?
It's a huge market, multi-billion-dollar market. We think there's at least 500,000 researchers and at least 80,000 labs that can use our system. Right now, we're in about 165 labs of the 80,000. We're a small company. We've had one salesperson for the last 9 years. We've had minimal money to develop things. We had one instrument that was run by a microprocessor. Things have really changed for us in the last year. We now have 6 salespeople. We've hired 5 salespeople in the last few months. We now have 4 new instruments that are ready to come out on top of the one we have. We now have a brand new instrument that is computer driven, not run by a microprocessor, has tremendous software. So things are much different today than they were even 6 months ago.
Any updates?
There are. At this conference, I gave a talk earlier today, and what I said in the talk is that we've received recently, in the last few weeks, 2 patents from China, and we filed these the patents elsewhere in the world, throughout the world. These patents have to do with a new process that we call ultra-shear technology. So in addition to our base business, which we think has a tremendous potential to grow, we have a second focus in the company now. This focus is in using a brand new technology that can make what we call nano-emulsions. Emulsions are where 2 or more liquids don't mix. These are emulsions, so you try to put chemicals in to get them to mix, like oil and water and oil and vinegar. We have a way using high pressure and high shear and valving that we've invented to, we believe, break particles down to nanoparticles, which allows them to be soluble in liquids that they're not normally soluble. So think about all the emulsions, the industrial lubricants, the food, the cosmetics, the pharmaceuticals that are emulsions, and it's hard to get them to be bioavailable because they're emulsions. They have chemicals in them, which we don't want. Imagine having them maybe chemical-free or maybe at 10% of the amount of chemicals that we call detergents and surfactants that allow them to mix. Imagine using much less or none and having them to be much more viable because they're water soluble. We believe that this new technology will allow us to take samples, liquids that are totally immiscible, can't mix, and allow them to be mixed and water soluble.
Rick, let's get your website out there for our investor audience.
Absolutely. www.pressurebiosciences.com. That's pressurebiosciences.com.
Ladies and gentlemen, Rick Schumacher, Pressure BioSciences, Inc. It's a publicly traded company, and the symbol is PBIO on the OTC markets. I'm Shelley Kraft. This is SNNLive. We're coming to you live from the Disruptive Growth Company Showcase 2017 in New York City. Rick, always a pleasure.
Always a pleasure. Thank you for having me on.
Good
really
to see
appreciate
you.
it.